Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
What if one of our most powerful allies in the fight against cancer wasn't some complex new drug, created in a lab, but rather something tiny, unexpected, a partner already hidden inside the very tumors we're trying to destroy? Imagine a bacterium. Just think about that. Not just living with the cancer, but actively helping to fight it.
That's quite a thought. And it's not science fiction, is it?
Not anymore, it seems. That's the really surprising idea behind some groundbreaking new research we're diving into today.
Yeah, this work is fascinating. It shows how certain bacteria that kind of often found living right inside and around tumors actually produce a specific molecule.
And this molecule does what exactly?
Well, it seems it can slow down cancer growth and make chemotherapy more effective, supercharge it almost.
That's a remarkable shift in perspective, isn't it?
It truly is. What's really interesting here is that we're talking about significant reevaluation of how we understand the complexity inside our bodies, especially with disease. So our mission in this deep dive is to unpack this discovery, understand the sort of intricate mechanics behind it, and explore what it might mean for the future of cancer treatment.
And where are we getting this info from?
We're drawing insights from a recent health news piece and, importantly, a detailed scientific paper. It was published in Cell Systems by a big international team. Researchers from the MRC Lab of Medical Sciences, Imperial College London, University of Cologne. Quite a collaboration.
Okay, so let's get into it. This idea of bacteria in tumors. Most of us know about microbes, on our skin or in our gut. That's familiar territory. But this feels different. Recent discoveries have shown that tumors themselves actually host these unique communities of bacteria.
Exactly. Scientists are calling it the tumor-associated microbiome. And it's a relatively new field of study. We're really just beginning to figure out the role these microbes play.
So for a long time, we just didn't know they were there or didn't think they mattered.
Pretty much. The focus was heavily on the cancer cells themselves, the host's immune cells, the surrounding tissue. That was the microenvironment picture.
Right.
The idea that bacteria could be active players in that environment, maybe even influencing how the cancer grows or responds to treatment. That's the newer, really exciting part.
And this team found something specific.
They did. A major breakthrough. Identifying a specific molecule made by these tumor bacteria that seems to have these powerful anti-cancer properties. It really suggests we might have microbial allies we just never knew about.
Microbial allies. I like the sound of that. So you mentioned this chemical conversation happening between the bacteria and the cancer cells. What did the researchers manage to overhear? What is this molecule?
Good way to put it. Yeah, their main goal was essentially to listen in on those chemical conversations. And they pinpointed this potent anti-cancer metabolite, a specific molecule produced by bacteria often linked with colorectal cancer.
Okay.
The molecule is called 2-methylisocitrate. But to keep things clear, let's just call it 2-methylocit.
2-methylocit. Got it. And how on earth did they find that specific molecule? I mean, bacteria produce loads of stuff, right? It must be like finding a needle in a haystack.
It really does sound like that, yeah. But their method was quite smart. They started with a really broad, large-scale screening approach.
Okay.
Testing over 1,100 different conditions in these tiny worms, C. elegans. You can do these screens relatively quickly with them.
Ah, the workhorse worms.
Exactly. And that allowed them to rapidly identify that a very common bacterium, E. coli one we all know, produced this 2-methylocit.
And right away they saw something interesting.
They did. This initial screen immediately showed promise. They found that 2-methylocit could significantly boost the effectiveness of a common chemotherapy drug.
Which one?
It's called 5-Fluorouracil, often just called 5-FU. It's used for quite a few cancers.
So the worms were like the first hint, the rapid test. But obviously you can't stop there. Worms are great, but they're not people. How did they confirm this wasn't just some, worm-specific thing?
Absolutely crucial point. They needed to validate it. So moving beyond C. elegans, the researchers used computer modeling first. They tried to predict 2-methylocit production in actual microbiome data from patient tumors.
Okay, so using real-world data pattern.
Right, and then they moved to lab experiments. They directly tested 2-methylocit's effects on human cancer cells grown in dishes. And they also used a fly model, Drosophila, specifically a model for colorectal cancer.
Flies, too. So worms, computer models, human cells, flies. covering the bases.
Exactly. And in both the human cell lines and the fly model, tumacet showed these potent anti-cancer effects. For the flies, it even extended their survival time.
Wow.
So yeah, the evidence really started to build up, suggesting this molecule had real potential beyond just the initial worm screen.
That is compelling. It almost sounds simple, like this one molecule does all that. How does tumacet actually work its magic inside the cancer cell? What's the mechanism?
It's fascinating, and the study shed a lot of light on it. It seems tumacet works by targeting a key enzyme, specifically an enzyme inside the mitochondria of the cancer cells.
Mitochondria, the powerhouses of the cell, right, where they make energy.
Precisely. So by inhibiting this enzyme, tumacet basically disrupts the cancer cell's energy production. It sort of messes with their metabolism.
Okay, cuts off the power supply in a way.
That's a good analogy.
Yeah.
And this disruption leads to DNA damage within the cancer cells.
So it hits them on multiple fronts. Energy and DNA integrity.
Exactly. And that DNA damage then triggers certain pathways, specific cellular responses that are known to slow down cancer progression.
So it's like a multi-pronged attack.
Yes. It weakens the cancer cells, makes them much more vulnerable. And here's the kicker. When they combine tumacet with that chemo drug, 5-FU,
the effect was even stronger.
Significantly stronger. Much better at killing cancer cells than either the tumacet alone or the 5-FU alone. That's what scientists call a synergistic effect.
Synergistic, meaning the whole is greater than the sum of its parts. 1 + 1 = 3 kind of thing.
Exactly. Leveraging the bacterial molecule and the existing treatment together for a much bigger punch.
That's incredible clever. So let's zoom out. What does this all mean for people listening, for the future of cancer treatment? This feels like it could be more than just an interesting lab finding.
I think it really could be. If we think about the bigger picture, it shifts our understanding of cancer. It's not just about our own human cells anymore. It pushes us towards seeing the body as holobiont.
Holobiont, meaning?
Meaning the human host plus all its resident microbes functioning as one interconnected biological system.
Right, acknowledging the passenger.
Exactly. And Professor Felipe Cabrera, one of the lead researchers, he put it nicely. He said, look, we've known bacteria hang out with tumors, but now we're starting to decode that chemical conversation they're having. Mm-hmm. He actually described tumacet as potentially a powerful partner for chemotherapy because it disrupts the cancer cell metabolism and weakens them.
And the first author on the paper, Dr. Martinez, what was his take?
He also expressed that sense of wonder, really. He found it truly remarkable that just one single molecule from a bacterium could have such a profound effect on cancer progression.
Yeah.
And he emphasized that this really highlights the complexity of biology, looking at it holistically, and stressed that, we are only scratching the surface here. There's likely so much more going on.
Which leads right into personalized medicine, doesn't it?
Absolutely. This discovery really underscores that future treatments might need to consider not just the patient's genetics, but also their unique microbial ecosystem. Your gut microbiome, for instance, is incredibly varied from person to person.
So my bacteria might make my treatment work differently than yours.
Potentially, yes. Or maybe we could influence that microbiome to help the treatment work better. Truly personalized medicine might need to factor in these microbial differences to really optimize therapies.
And it gets even more interesting because the researchers didn't just stop with the natural molecule, did they? They actually tried to improve on it.
They did. This is where it moves towards drug development. They collaborated with medicinal chemists to tweak the structure of the natural tumacet compound.
Yeah.
And this synthetic modified version turned out to be even more powerful at killing cancer cells in their tests.
Wow. So taking a hint from nature and then engineering it to be even better.
Precisely. Imagine designing completely new drugs based on these natural microbial products, but optimized in the lab. It opens up a whole avenue for drug discovery.
It really does. But we should probably add a note of caution, right? Are all microbial molecules helpful like this?
That's a very important point. No, they're not. It's crucial to understand that microbial metabolites can be, as the paper notes, a double-edged sword.
Meaning some can be bad.
Yes. While tumacet looks promisingly anti-cancer, other compounds produced by gut bacteria, like certain secondary bile acids or maybe some metabolites from tryptophan, have actually been linked to accelerating tumor growth, possibly by increasing inflammation.
Okay, so it's not just any bacteria, any molecule. It's about finding the right ones.
Exactly. This research is about harnessing the beneficial side, finding those specific helpful molecules like tumacet, and figuring out how they work and how we might use them.
Which brings us to the next logical step. Every exciting study like this has limitations, things that need more work. Understanding those is key, right? So what are the caveats here? What's the path forward?
Yeah, the researchers are quite upfront about this. They acknowledge that at this stage, it's hard to say definitively how well these findings will translate directly to human patients in the clinic. Generalizability is still a question mark.
Why is that? What are the main limitations?
Well, partly it's the data they used for the microbiome analysis. They used existing published data sets which weren't collected specifically for this study. So while useful, it's not quite the same as tracking tumor-associated production in real time within the patients being studied.
Okay, so the data source is one thing. What else?
And then there are the model systems. A lot of the really crucial initial work, the mechanism stuff, was done using those invertebrate models, the worms and flies and the human cells, in lab dishes, in vitro.
Right.
And while those are incredibly valuable tools for discovery and figuring out how things work at a basic level,
They aren't perfect replicas of a whole human body.
exactly. They might not fully capture the incredibly complex dynamics, the immune system interactions, everything else that happens in a real person undergoing treatment. The human body is just way more intricate.
So more research needed, basically.
Absolutely. These limitations really highlight the critical need for more experimental work, particularly moving into mammalian models like mice and using other methods to really confirm if these exciting findings hold up and could eventually be applied safely and effectively in routine clinical practice.
We're still in the relatively early stages then, but it sounds like a very promising start.
Very promising, yes.
Okay.
The foundation they've laid for future work, potentially leading to clinical trials down the line, seems really solid.
Okay, so let's just recap this deep dive then. We've learned that hidden within tumors, there are these communities of bacteria. And one molecule they can make, this 2-macet, seems to act as a potent anti-cancer agent, especially when you pair it with existing chemotherapy like 5-FU.
That's right. It disrupts the cancer cell's energy supply and damages their DNA, basically weakening them.
Opening up a whole new potential front in cancer treatment using these tiny microbial allies.
It really does.
Well, yeah.
And this deep dive into our microbial partners, it really makes you wonder, doesn't it?
How so?
Well, if we can identify and maybe even engineer these bacterial products, how might we revolutionize personalized cancer treatment, moving beyond just, fighting the cancer to maybe allying with some of the life already within us?
That's a fascinating thought. What could the future of truly personalized medicine look like if we start factoring in not just our own genes, but also these huge, complex ecosystems of microbes living inside us?
Exactly. What other secret conversations are happening? How many more natural, powerful partners are just waiting to be discovered right under our noses, or rather inside our tumors?
It's a powerful reminder, I think, that sometimes the most revolutionary solutions aren't in the places we expect. They might be found in these tiny, unexpected corners of biology.
A fascinating path forward, for sure. A whole new generation of potential therapeutics may be inspired by nature's own chemists.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.